You were talking a lot. The training. You recognize me by their voices now.
The Queens College Noise SciTech Teams program prepares highly qualified STEM teachers from underrepresented populations in an effort to create a sense of community, science literacy, and engagement with science for all ages. But I'll let our teacher candidates demonstrate the pedagogical techniques they've learned in our program. Educational technology is essential in today's classrooms. Therefore, we emphasize the strategic implementation of technology to maintain an environment of engagement and learning that stimulates science inquiry. Gaming, real-world data, and mixed reality technology are just some of the ways our teacher candidates integrate theory and practice and put their creativity on display.
Hello and welcome. I hear that you're interested in the new meteorology position that's just opened up.
In addition to using technology in the classroom, our candidates use technology to enhance the materials they create for their students. We've also promoted a STEM teacher network where our alumni are actively engaging with our teacher candidates. We believe community engagement is a foundational tool to enhance science literacy. Therefore, we provide ample opportunity through conferences and cross-departmental demonstrations for our teacher candidates to practice their art of teaching.
Okay, that tells me I should be worried about this toothpaste mixture.
All right, 3, 2, 1.
A unique aspect of the SciTech Teams program is the emphasis on a variety of pedagogical competencies to produce well-rounded STEM teachers. Our teacher candidates learn outdoor education as well as globe protocols at our field station at Black Rock Forest. This excursion has proven to be critical for cohort building and generating new avenues for science interest. Informal science education provides great entry points for young students to explore and engage with science. Our SciTech Teams participants have an opportunity to also explore and engage with this entry point at the New York Hall of Science. Our partnership with the Hall of Science offers our teacher candidates training with an emphasis on inquiry and creativity that they can bring back to the classroom. A combination of strong educational technological pedagogy, practice with outdoor and informal education, as well as training with the next generation science standards, has led to a successful science educator community that continues to enhance their content knowledge, pedagogy, and engagement for the benefit of their students.
Okay, thank you. Good morning, everyone. Welcome to the 4th annual Reimagining Science and Technology Education Conference at Queens College. I'm really happy to be able to say that back in person, to see a room full of people after doing the conference online last year. So thank you. My name is Salvatore Garofalo. I'm a faculty member in the Science Education program at Queens and a member of the Noise SciTech team. Along with the rest of the team, Dr. Stephen Ferenga, Dr. Gopal Subramaniam, Dr. David Lawrenson, and Dr. Natalia Holtzman, we wish to welcome you to this day that celebrates science, technology, and its important intersection in all of our classrooms. We would also like to thank and acknowledge the support from the Dean of the School of Mathematics and Natural Sciences, Dr. Daniel Weinstein, the Interim Dean of the School of Education, Dr. Dana Fusco, and the Chair of the Secondary Education Youth Services Department, Dr. Eleanor Amor Thomas. And thank you to all of you, those of you that are here, those of you that are joining virtually, and all the schools that we're broadcasting to. Good morning. I'd now like to introduce our keynote speaker. Dr. John Dennehy is a virologist who's interested in all aspects of virus biology, ranging from virus molecular biology to virus ecology and evolution. Dr. Dennehy earned a PhD at Clark University and was a postdoctoral fellow at Yale University and the State University of New York at Albany prior to joining the faculty at Queens College in 2007. Research in the Dennehy laboratory employs bacteriophages, rotaviruses, and coronaviruses as model organisms to test predictions generated by theory and mathematical modeling. They probe how viruses evolve at the molecular level to best exploit host cells and propagate a host populations. Additionally, they explore gene expression regulatory mechanisms to determine how processes such as virus infection, assembly, and life history event timing are controlled. Other topics of interest include the biology of virus collective infection via extracellular vesicles, virus coevolution and exploitation of hosts, phage therapy, and virus systems biology. Dr. Dennehy has published over 50 articles, book chapters, and conference papers on virus biology. He is also a senior editor of microbiology resource announcements, an associate editor for virus evolution, and a curator for the American Society of Microbiology's COVID-19 research registry. We are very excited to have Dr. Dennehy here with us today. So please join me in welcoming Dr. Dennehy, our keynote speaker.
It's my great pleasure to be here today, and I'm fortunate to see some of my former students in the audience and online at home. As you know, I'm a professor at Queens College. I tremendously support the education mission of high school students in our local communities. We often have them come to Queens College and participate in research in my classes. And I'm very encouraged to be here to share this mission with you. And additionally, I want to talk about SARS coronavirus and what we've been doing here at Queens College for the past 2 years, almost 2 years now. Basically, what we want to do is measure the levels of SARS coronavirus in wastewater. You might be surprised, this is actually a source of viruses. Not everyone, but many people get infected with SARS coronavirus, shed this virus in the feces, and the viral RNA will persist for a long period in the wastewater. So we're able to collect it at the source, the wastewater treatment plant in New York City, the 14, and we can assess whether SARS coronavirus is present, and if present, how much is there. And this gives us a way to do an unbiased sampling of the entire city. Most of the clinical sampling requires someone to come into the clinic and get tested, usually with a nasal swab. So if you don't get tested, you don't know you're infected. And this is especially problem when now that people are testing at home, we can't really get great data on how many people are infected in the city. So this wastewater sampling allows us to surveil the entire 8.5 million people in New York City. Plus, we can do it at a cost that's much, much cheaper than would be the case if we are sampling from individuals. So this is developed in a partnership with the New York City Department of Environmental Protection, and they are responsible for making sure we have clean wastewater that gets released into the harbor. Occasionally with storms, we have overflow of the system, and then sewage goes into the harbor, and they're responsible for making sure that our beaches are safe and so on and so forth. Additionally, we partner with the New York City Department of Health and Mental Hygiene. They will be the main people who are tracking any outbreaks of any epidemic in the city. And of course, with SARS coronavirus, they took the responsibility of monitoring the number of cases in the city. The DEP maintains a microbiology lab at Newtown Creek. You might have seen this if you're driving down the LIE and you look up to the left or the right, depending on which direction you're traveling. There are these large silver composters which they ferment the wastewater waste, and they're quite obvious when you look at them. I think it's just before you get to the Midtown Tunnel. Anyway, the DEP run 14 different treatment plants in each one of the boroughs. You can see this map here. There's several locations in Queens. Right now, we're in a location called TI, Tallman Island. So the general principle, basically, people said SARS coronavirus in their feces. This goes into the waste stream. What we do is we recover that SARS coronavirus from the wastewater, and that basically looks like this, a virus particle. There'll be billions of these in the wastewater, and notably, they have what's called a spike protein, which helps them to attach to human cells. Once it gets into the wastewater, no one has recovered infectious virus, so it appears that that process somehow inactivates the virus, and it's not infectious. So it's quite safe when you bring it into the lab, but nonetheless, we pasteurize it 60 degrees, 60 minutes, and that inactivates the virus and prevents it from infecting any of our employees. We recover the virus RNA, we convert that RNA into cDNA. For those of you who are taking my microbiology class, you might be familiar with that process. From that cDNA, we can then use what we call real-time quantitative PCR, which will tell us how much viral RNA is in the wastewater. And then additionally, this just gives you the information about the number of virus particles in the water. We do sequencing to identify what genotype of the virus is present. That can tell us information whether different variants of concern present in the wastewater, such as the Delta, the Alpha, the Omicron, and all that kind of stuff. And we actually developed a unique approach called targeted sequencing, which I'll talk about in a bit, because of the challenges of sequencing SARS coronavirus from wastewater. But the RT-PCR protocol that I mentioned, this is able to measure basically the concentration of virus in the water. And there's a very good correlation between the concentration of viruses in the wastewater and the number of clinical cases that we see in the hospital. That said, this relationship is becoming worse and worse. It's not because the wastewater sampling is getting any less efficient, but rather more and more people are testing at home, or they are asymptomatically infected and not going to hospitals and getting diagnosed. So we actually feel now that the wastewater virus concentration is a little more accurate at reflecting what's actually going on in the community than is what you see in the New York Times when they say there's 1,000 cases per day and over the last 7 days or something. So importantly, what we do, this has always been our intention from the very beginning, was to monitor SARS coronavirus evolution. And when we first started doing this, most people were not interested in monitoring SARS coronavirus evolution. The belief was, among many virologists even, but the SARS coronavirus wasn't going to evolve very quickly for technical reasons. And we applied for NIH funding, and they said, we don't even think you can sequence virus from wastewater. Don't bother us. So what changed was back in November, December 2020, when all of a sudden we started hearing about the Alpha variant, Beta variant. And then suddenly there's a big interest in monitoring SARS coronavirus evolution, how these kinds of changes entail different ability of the virus to be transmitted between individuals, and also their ability to cause virulence in patients. So we wanted to make sure we're observing those things. And additionally, as the virus starts infecting people in the population, or people start getting vaccinated, the virus can also respond by mutating to evade antibody-mediated immunity. And knowing what variants are currently infecting the population will also influence the selection of vaccine if we're able to update the Moderna or the Pfizer vaccine. That's the information that they need. The clinical sampling is able to do this. We can get this information from nasal swabs, we can sequence the virus genome from that and be able to tell what variant is present. But this is relatively low throughput. It's very expensive per sample, it's a couple of $100, and it's biased by infection type. So only people who generally go to the clinic are those that are getting sequenced. So if you're asymptomatic or you're testing at home, you might not be getting that information. Additionally, it's biased by access. Many people who without insurance or undocumented immigrants or different marginalized individuals are not getting tested like the wealthier people in the community. And that is actually a factor across the entire world, not just in New York City. We have very good sequence representation from the United States and from the United Kingdom, but other places in the world like Africa, Southeast Asia, we don't have as much information. And one big thing that came from Africa was the Omicron variant, and it just appeared out of nowhere, kind of fully mutated, and that took a lot of people by surprise. And part of the reason we missed it potentially was we were not sampling in that region. So the sequencing of virus from the wastewater is able to overcome some of those limitations. Like I said, it's much cheaper, and you can sample from an entire community at the same time, and it's much more feasible to monitor virus in places like sub-Saharan Africa. And we're actually now partnering with a group from South Africa to show them how we're able to do this targeted sequencing. South Africa is a big concern. They have a very large population of HIV positive individuals, and I'll talk later about why that's important. But when we first started doing this sequencing, we tried what everyone else did, which is to sequence the entire genome of the virus. And this didn't actually work out so great for us. This is probably one of the worst examples, but a very poor quality genome that we were able to recover. What the spikes here mean is that we get what we call good coverage at this location. We get a lot of sequence, we're pretty confident about what the genotype is there. But in the other regions where you don't see anything, those are gaps where we don't get any sequence at all. So we lack a lot of information about what kinds of mutations are present in the genome. And this especially applies to what we call the receptor binding domain. When I showed you the figure of the virus earlier, it had the spike proteins. Now those are going to bind to the host cell. And the spike protein is basically in this region here. The important part of that spike protein is the part that actually makes physical contact with the receptor on the host cell. And that's what we call the receptor binding domain. So here we can see this receptor binding domain binding to the ACE2 receptor on human cells. This is the primary determinant of what animals can be infected by SARS coronavirus, and additionally, what cells can be infected. And additionally, about 90% of all neutralizing antibodies that are generated against SARS coronavirus target this region. So we know it's very, very important. Most SARS coronavirus 2 mutations that appear are appearing in the receptor binding domain, and the sequencing of this area can be extremely informative of what variant is currently circulating, and also to predict what might happen in the future. So for this reason, we decided, well, if we can't get the whole genome, maybe at least we can get this very important region. So we tried it, and we amplified this region, and it was tremendously successful. This is that same kind of figure I showed you with the spike protein receptor binding domain. We get extremely high coverage relative to what we got when we did the whole genome sequencing. So from this, we're very able to see what mutations are present in the community. And also because we're targeting just a small region, we can tell if there's different variants circulating. where the whole genome sequencing, you might not be able to tell whether a mutation in the envelope protein is co-occurring with a mutation in the spike protein. They could be two different variants. But with this approach, we're able to tell that the mutation occurring within the same virus. So here's a here is actually what we see in our data. I've kind of compiled it into like very busy visual. But let me explain what's going on here. Each one of the rows is a different genotype of virus that we've recovered. So there's quite a great deal of genetic diversity that's being recovered. The boxes over here represent whether we detected that virus at that time, because we can trace it down to the date where it was detected. And the color of the box indicates what the frequency of that variant was at that time. Additionally, there are different colors of these boxes here. And what they mean are the orange boxes mean that the type or genotype has the same mutation at that loci that Omicron does. And then the green boxes indicate that the variant has a mutation at a loci that is mutated in Omicron, but it's not that same exact change. And then the black boxes are just other mutations that are occurring at the same time. So, over time, you can see how the virus is actually evolving and it's expanding in genetic diversity. That's one very interesting thing that we're seeing is that they're looking for different solutions to help them better infect patients. Incidentally, this particular variant, should I hide that? Up here to the title, so it's being blocked out. But anyway, this particular variant is found in Coney. This particular variant was found in southern Staten Island at a wastewater treatment plant called Oakwood Beach. There are other variants around the city. This one was found in Owl's Head, which is southern Brooklyn. This one was found in Coney Island. This one was found in Bowery Bay, which is northwestern Queens, like Astoria, Elmhurst, Corona. And this one was found in this is Ward Island. So this is the Western Bronx and northeastern Manhattan. And then finally, Palmer Island is the most recent variant we've seen. I want to emphasize something here. All of these variants that we see in the wastewater were never detected in patients. And we don't quite understand why. It's very curious that we see, and there's quite a bit of virus being detected some of the time, and sometimes it would even outnumber the currently circulating virus, whether it was Delta, Alpha, Omicron. But whatever is setting these viruses into wastewater was setting so much that it outnumbered the common variant of concern that was circulating in the city. So we have this really big mystery of where these sequences are coming from. New York City poses a pretty big problem in trying to trace the origins of these viruses, which is so giant. We attempted to do this for the variant we saw in Owl's Head, which is southwestern Brooklyn, and we did what we call subsampling. So instead of going to the wastewater treatment plant to get our sample, the DEP actually went to manhole and collected a sample there and did it in a way that we could narrow down a smaller area where the variant was coming from. And this worked tremendously well. And we were able to narrow it down to a small region of Brooklyn. Problem was that small region of Brooklyn still contained several 100,000 people. and it still contained several hospitals, several nursing homes, animal shelters, and it was kind of, very difficult for us to further narrow it down in such a big city like this. So what we did, we decided to reach out to our colleagues all over the country and all over the world and ask them for samples from their own location. Many of them had also been sequencing, but they had applied the whole genome sequencing strategy, which we didn't find very effective, and they weren't seeing any cryptic variants. But when they sent their water to us, or their RNA to us, and we tested them, we were able to detect variants in several different locations: New York, New Jersey, Wisconsin, Missouri, California, and all of the other states. I think at this point, I haven't updated the figure, but we tested all the blue states as well. So the states in blue and red are places where we tested for cryptic variants, but we didn't identify anything. Additionally, we tested samples from England, Italy, and Argentina, and we hope to get more over the, in the near future that we can also test. And to further emphasize how different these things are from the currently circulating variant, this table kind of shows different mutations that are happening in the receptor binding domain and whether or not they were present in any of the variant of concern. The ones in green indicate that these mutations were appearing in variants of concern that appeared before Omicron. The ones that blue were very, very rare prior to Omicron, but did appear in the Omicron variant and are now exceptionally common. And then the ones in yellow are just exceptionally rare, period. There's one I kind of highlighted earlier, Q498Y, and that one had never, ever appeared in a patient before until just fairly recently in February when we first saw it. And even today, there's only about 16 patients worldwide that had that particular mutation. The numbers are over here, just kind of showing you how rare these things are. Now, to keep in mind that we're sequencing at an unprecedented rate. There's something like 9 million sequences in a global database we call GIS-AID. So out of those 9 million, we had 12 that had that Q498Y mutation. That's exceptionally rare. But that Q498Y mutation appears in many sewage sheds widely spread about the world. So it's clearly important. The other thing we know, based on looking at some of the bigger sequences we've been able to recover, is that most of these variants originated down here very early in the pandemic. We think some of the variants of concern from New York City, I shouldn't say variant of concern, I should say cryptic variant that we discovered in New York City, we believe they probably arose in the first wave between March and May of 2020. That's also the case for some of these other ones in different cities. Now, this chart kind of shows how many mutations these things have, and we can trace back in time when they must have arisen. And you can also see different branches or other variants of concern. And they also seem to have arisen quite early in the pandemic. So this begs the big question. It's been bugging us for well over a year now. Where the heck are these things coming from? Come up with 3 different hypotheses. It could be unsampled infection. This means that there are patients walking around, they're completely normal, they just haven't been tested. And they contain the oddball variant, whereas people who are tested are the ones that have normal ones. And there's no reason why there should be that way. Because New York City is testing about 10% of all the clinically diagnosed cases. Tens of thousands of people have had their genome sequenced in New York City, and we've never seen these variants before in a clinical sample. So it's extremely unlikely we're just missing those patients by chance. The second hypothesis was that it's coming from some sort of animal reservoir. And this, when we first saw this data, that was our first thought, that it's got to be an animal reservoir, because we didn't believe that these patients are being missed if it's in fact in a human or coming from a human. And when I first saw that mutation called Q498Y, I didn't see it in the global database, so I Googled it. I just typed in my computer Q498Y, SARS coronavirus, and the first thing that comes up was a paper from Ralph Barrett's lab in North Carolina. What he had done was that the original Wuhan strain of SARS coronavirus could not infect human cells. So what they wanted to do was they wanted to develop a model that they could experiment with to see how SARS coronavirus behaved in human cells. So they actually had to, no, I should say, I'm sorry, not human cells, mice. They wanted to find a virus that could infect mice. Now, the original Wuhan strain cannot infect mice. So they adapted the virus such that it was able to infect mice. Key mutation that allowed the virus to infect mice, Q498Y. So once we saw that mutation, and once I saw that paper, and I found a couple of others demonstrating the same thing, I said, it's got to be rats, because rats everywhere in New York City. And they're probably going down the sewers and drinking the water, right? So what we did was we created what we call pseudoviruses. Now, these are not actual viruses, but they can express the spike protein, and you can test whether that spike protein can bind to cells containing either human, mouse, or rat ACE2 receptors, the receptors the virus binds to. In the control, we don't see any binding. But those viruses that contain the spike protein from one of our variants were able to both infect human cells very well, but also they were able to infect mice cells and not mice cells, human cells expressing the mice receptor and also rat receptors. So this demonstrates that the viruses that we're pulling out of the sewer should be able to infect rodents. So, you know, this is pretty convincing evidence for us, but we also wanted to know what animals are contributing to our sewers. And so we did something where we collected RNA from the sewers and we sequenced an area of the RNA that is actually the ribosomal RNA protein, which helped to create ribosomes. And we're able to map that to different animal species. Now, these are the most common animals that we were able to detect in the sewers. Now, they contain some interesting hits here. Cows, pigs, sheep. You wouldn't expect them to be wandering around the city, of course. So what we're actually detecting there is what people consume, beef, pork, chicken, creating it out in their feces. We're actually able to tell what food people are eating in New York City, which is kind of cool. Additionally, outside of those food animals, we're able to detect rats, cats, and dogs. So it's quite well known at this point that rats, cats, and dogs all can be infected by several strains of SARS coronavirus, but do know that the dogs and cats, although they can get infected, they do not seem to be able to spread it to other animals or even to humans. There's sort of iffy data there, but for the most part, it doesn't seem like a likely pathway. Also, it's not the case that we can often see large congregations of animals. You know, there's no cat parks out there. We considered feral cat colonies. We considered animal hospitals, but none of our samples really came from those kind of places were positive. Hence, we thought that rats were the most likely suspects. They both significantly outnumber the number of cats and dogs in the city. They do have quite a bit of access to the wastewater. They're able to go down into the sewer, and it's plausible they could be shedding their feces down there. And we tested some of the feces, and we did see that they do get infected by SARS coronavirus. Said every time we sequenced the virus that came from SARS coronavirus, it always turned out to be the contemporaneous strain. We didn't see this cryptic variant appearing in rats, so we never got that smoking gun. And many other labs have done some similar things, and they either did not detect SARS coronavirus or they weren't able to detect any weird variant. So that leaves us with a third hypothesis. These things are coming from long-term infections of immunocompromised patients. This is something weird, just an odd curiosity in the beginning. Is that some patients that have cancer or were getting organ transplants or have HIV, they were getting infections that they could not eliminate from the body, even with really strong treatment with monoclonal antibodies and antiviral drugs, and the infection would persist in the patient. I think the longest one I've heard is 14 months. That's an extremely long time to be infected by a virus. So what we think is happening is that the virus is infecting the patient, it's never been cleared, and it continues to evolve over time. And the body tries to put up a defense. It's not a very effective defense. Nonetheless, it's mild enough that it will actually pose some selection pressure on the virus to evolve adaptation or mutation that will allow it to evade antibody-mediated neutralization. So other point is that we see in these clinical cases, the virus evolved at a much, much faster rate than infection of a normal patient, even those with so-called long COVID. We tested some of our variants, not the actual virus, the pseudovirus, and we found that many of our variants, they're called in this figure WNY134, and this figure here is just showing how well this variant is being neutralized by different antibodies. These are the monoclonal antibodies produced by Eli Lilly, Regeneron. If the virus is being neutralized, you'll see a big drop. This is the ancestral sequence. So you see the big drop in the detection of this variant in the figure. But the ones that are not being neutralized just stay straight. In the second part of this figure, we also tested it with serum obtained from either patients that had recovered from virus or that had been vaccinated. And this is kind of the opposite of this figure, but if the virus is being neutralized, you should see a drop in the neutralization efficiency. And that's what we see. We see much greater neutralization efficiency in the wild type versus the variant that we discovered in the wastewater. So it seems that these variants are highly resistant to monoclonal antibody and also to antibodies generated naturally within the body after being infected or vaccinated. In the ensuing time since we made the discovery, more and more evidence has been accumulating that some of the variants of concern may have actually arisen within immunocompromised patients. The Alpha variant disappeared late in 2020, and it kind of disappeared out of nowhere to the people monitoring it in Great Britain. It is related to the ancestral sequence, obviously, but they are not able to find any intermediary forms before you find this Alpha variant that has several mutations allowing it to evade antibody-mediated neutralization. Additionally, the Omicron variant appeared extremely heavily mutated in South Africa or Botswana or somewhere like that. And again, it appeared out of nowhere. They were not able to detect any intermediary forms leading up to that heavily mutated virus that started appearing in patients. So what the hypothesis is that these variants were infecting immunocompromised patients who were not getting tested. It could be untreated HIV patients. And they're able to see one of the extremely early cases of Omicron in South Africa was actually found in an HIV positive patient who was not receiving antiviral drugs. So the big question for us is, we know that these things are appearing. They're appearing in patients. We know that they're appearing in patients with HIV and organ transplant patients and cancer patients and anyone with a suppressed immune system. Most of us virologists think that it's only a matter of time before the next variant of concern appears. That variant of concern will be named pi, which is the next letter in the Greek alphabet after Omicron. So we believe that variant is probably out there right now in a patient who's not being treated and is likely to be extremely highly resistant to antibody-mediated neutralization, potentially to antiviral drugs. It could be more transmissible or more virulent than Omicron. It's not necessarily the case that we will always evolve less virulent forms of viruses. We can evolve more virulent. Delta was more virulent than Alpha, for example. So we strongly believe there needs to be a concerted effort to find these patients who are infected and to cure the virus in them, or at least to isolate these patients so that they're not transmitting pi to the general population. This is kind of difficult to do without stigmatizing specific groups. We know that there's a lot of racism that was associated with the original SARS coronavirus and attacks on Asian people who are believed to be spreading the virus. Unfortunately, we can envision some sort of similar scenario where, ignorant people in the population will attack HIV patients because they fear they're the next source of the virus. So this really needs to be done without stigmatizing subgroups. Difficult to do. That's all for my talk, and I just want to thank the members of my lab. Most of them, all of them are Queens College graduates, and additionally, I want to thank Monica Trujillo. Many of you might know her. She's a professor at Queensborough Community College, and she's been instrumental in driving the project forward. And also I have a collaborator at the University of Missouri who helped me do the sequencing, named Dr. Johnson. I think I probably have some time for some questions. Have anything you'd like to ask?
How sure are you about the immunocompromised patients?
How
What
sure?
about the normal patients? Could they also be, have you tested in the real
Well, the
infected
normal
patient?
patients seem to recover fairly quickly, so there's not enough time for the virus to really evolve within a patient. And also when you transmit the virus from one patient to another, it goes through a very large bottleneck, meaning that only a few of the particles within the patient's body actually get spread to the next patient. When you sneeze, there's a little droplet, presumably, that will travel through the air. And that it's thought that that's really limiting the ability of the virus to evolve. The immunocompromised patient, we do have evidence that at least one of the cryptic variants that we've seen has been found in a patient. I really can't say where, but we do have a connection between cryptic variants in the wastewater and a patient who had HIV that was untreated. And unfortunately, that patient passed away before we could do further testing. But
is it possible that Omicron is less severe in terms of symptoms because it showed up in immunocompromised patients as Delta?
say again?
So Omicron isn't as severe or lethal as Delta? Is it because is it possible that it's because it showed up in immunocompromised patients? Yeah. Never mind.
It's in a way it's asking like, why is Omicron less?
Why are the
less-
symptoms
yeah.
less
Why
severe?
Omicron last year?
And is it possible it's because it showed up in?
Omicron, I think, is selective for transmission and also to breakthrough in antibody-mediated neutralization. People who are getting infected with Omicron tend to already have been vaccinated or have been infected. So there's relatively few people who unvaccinated and uninfected. We haven't actually noticed any significant spike in virulence, but I suspect it's probably more the case that Omicron, we just know how to treat the virus better than we did in the beginning. When the pandemic first started, there was a huge, I don't know if you're aware of this, but there's a huge debate in the medical community. How do we treat these patients? And a lot of the things they did at first were actually counterproductive. They ventilated too early, for one thing. And by putting people on the ventilation, that seemed to tip them over the edge and ends up with them dying instead of they perhaps have waited longer, they may have recovered. So we definitely significantly improved our ability to cure infections in patients. Plus we have newly approved antiviral drugs. We have monoclonal antibodies, which were not very common in the beginning. So I think those are actually driving down the death rate quite a bit.
I was just like asking, the reason why I was asking about that question was, we were always looking for this virus to multiply in our respiratory system. But what if it launches somewhere else? And then it is actually mutating over there. We are not observing, even in the normal patient. That's what.
So, you know, everyone thinks this is a respiratory virus, but there's actually evidence that it infected guts as well. And we've actually seen evidence that there can be a different variant infecting the respiratory tract and gut. So you have two different strains depending on where you look in the body. And it's also potentially the case by sampling your with the nasal swab, you're seeing one of those variants, but unless you sample the feces, not seeing both of the variants. Infection can persist much longer in the gut than they can. They're cleared from the respiratory tract relatively quickly. In the gut, stay much longer, and often the case that could involve more. In the back.
Saliva sample. The difference with saliva samples as opposed to the nasal sample?
You mean a fecal sample?
No,
No.
Oh, the mouth. Yeah.
saliva.
So this is actually something I think is a big missed opportunity, is that there's so much emphasis on nasal swabs. I think it reduces the, you know, people don't want to be poked in the nose. It's not pleasant. Whereas if you just spit in a tube, just is effective at detecting SARS coronavirus. That's actually what we do on campus. If you've been randomly tested, or you know, you spit in a tube, that is tested for SARS coronavirus, and there does not appear to be any difference in the effectiveness of each mechanism. Unfortunately, the medical community is centered on the nasal swab, which are effective, but they're a lot more, you know, painful than just spitting in a tube. And I think that reduces, you know, people avoid getting tested for that reason. They don't want the unpleasantness of being probed in the back of the nose, right? Not so much. I've been advocating from the beginning that we should be, using spit test instead of nasal swab, but, what I would really like is that we do more fecal testing, so-called butt swabs in China. If you think you have a hard time getting people to do the nasal swab, let me tell you.
Dr. Dennehy, what made you decide to use the parts of the DNA, parts of the RNA you were looking at?
Yeah, basically we were getting bad data, and we couldn't. We were interested in seeing changes relative to the ancestral wild type. The sequence we were covering was the same with the wild type, but we suspected there was evolution occurring because we knew there was the alpha variant and the beta variant, and we weren't able to see them. So we said, we got to change our approach. And if we just target the receptor binding domain, we'll be able to identify the variant and discriminate it from the wild type.
And is that where most of the testing is now? Doing what you are doing, your type of testing?
Yeah, so that's actually increasing numbers of people coming around to using this approach. We've been asked to share a method. The Bill and Melinda Gates Foundation wants to apply this to South Africa, kind of the collaboration I mentioned. Public Health Laboratory in New York City, Pandemic Resource Laboratory. They're the main group of people who sequence SARS coronavirus in New York City. They're also receiving wastewater, and they're using our approach instead of using the whole genome approach for wastewater. It seems to work fine in clinical samples because clinical samples tend to be much purer. There's a lot of other stuff in wastewater besides SARS coronavirus, right? There's a lot of detergents, there's industrial chemicals, there's, whenever someone cleans the house, they shampoos are going down the drain, that sort of stuff.
Just sequencing, for the part that you look at, are you able to predict how virulent it would be without actually having data?
It's really difficult to do because you can see the ability. Hypothetically, if we had a BSL-3 lab, which we don't, but people who do could test it on cell culture, or they could test it in mice. But you can't take a variant, test it on humans, right? You can only infer from people who are getting infected with that variant. Since we don't have actual cryptic variant, have never been able to recover one of these things, we can't even test it on cell culture. We can generate a pseudovirus, but that's not the real thing, right?
Could they make a vaccine if they knew that the future variant was coming?
Yeah, so that's something they're very interested in doing, is that we know there's loci that are getting mutated, and they can use that information to generate vaccine that can account for those different mutations. And those mutations are associated with antibody-mediated neutralization. We can generate a vaccine that evades the evasion.
We have time for one more question, if anybody.
Yeah, there's about the rate of mutation, like how fast any number.
Say again?
How fast the mutation is.
So they're mind-bogglingly fast. They're happening at a rate that I've never seen before in my life. We thought this would be a very slow evolving virus. It's an RNA virus. RNA viruses are supposed to evolve rather quickly, but SARS coronavirus is an unusual RNA virus in that most RNA viruses don't have the ability to proofread their own replicated genome. Humans do, bacteria do, DNA viruses do, but RNA viruses have a much higher error rate, mutation rate, because they can't proofread. SARS coronavirus the member of a group that does have the ability to proofread their own genomes after it's being replicated. So we did not expect to see so many mutations accumulating. And that's just basically a testament to how many infections are out there. More and more people are getting infected by this virus. It numbers in the billions of people. And each patient is generating trillions of viruses during the course of an infection. So much opportunity for the variant to acquire new mutation.
That's where you go back from the alpha, go with the,
Can you go back?
like the origin of the virus itself.
Back and maybe.
Yeah, so where could have started? Where or how or what is the origin of this alpha?
Oh, sure. That's actually one of the slides I showed is that we think the variant must have arisen very early in the pandemic, and because it has mutations that were only seen back then. There were lack of mutations, I'll say. In China, the virus had what we call the wild type sequence. Everything from, now the first infections happened globally in Iran, Germany, Italy, Spain. All of those viruses had a new genotype that was very different than what we see in Wuhan. What we see in the cryptic variant in New York City is something intermediary between those variants that we're seeing in Italy, Iran, Germany, and the Wuhan. So they must have arisen somewhat before those Italy, Iran, and Germany sequences became common. It still seems to be pretty effective against the Omicron variant. So there's no immediate concern, but I predict it's probably going to be a new vaccine released at some point. Probably, I would anticipate before fall, because the winter season is when we see the cases start to kick up. In New York City, I think every September we started to see increases. I mean, it's not that much time. It's been 2 years, right? But each September we see a summer lull, and then we see an increase September through January. So I anticipate they're going to release something to handle that next wave, hopefully by the fall. Something that will anticipate future variants that are appearing. Unless we don't see any new variant appearing, then maybe they'll decide to hold off on that. At the current vaccine does seem to be quite effective against Omicron, some of the Omicron derivatives.
There is one question in the chat. It says, with these mutations going on, do you think there's a possibility of another lockdown, even though people are getting vaccines?
I don't see another lockdown happening. I think that ship is sailed. I think Dr. Fauci has announced that the pandemic is over.
Right.
So just learning to live with the virus at this point. I think anyone that is currently vaccinated and had a booster, very well protected. And that's why you don't see me wearing a mask anymore. I'm not, the treatment for the virus, if you do get infected, is very good treatment, and most healthy people should recover quite quickly. There are people who have quirks of the immune system who tend to be much more sensitive and can get a really bad viral infection, but those people are extremely rare, and I'm just hoping I'm not one of them. So.
Okay. Thank you very much. Really appreciate it.
Personally, thank Dr. Dennehy for explaining both science peers, science educators, and scientists, and also our people at home. Dr. Dennehy, if people like you shared more of this information, maybe we wouldn't have the misinformation out there. So greatly thank you for taking your time, coming here to speak with us today. Greatly appreciate it.
I don't get a chance to say this very much, but I often invite, well, people reach out to me, middle school groups and high school groups. They often come to visit my lab and visit the science building. In the future, if you ever a teacher, you want to show your class what a science lab looks like, you're welcome to come by.
I appreciate that.
Yes, thank you. Okay, so it's amazing there's more to do after that amazing keynote. But the next part of our conference today, our workshop. So all of you should have received an email late last night about which group you are in. So you could further check your email, or we're flexible here. So just so you know where everything is, if you were placed in group A and B, you're going to start with programming with Arduino. Thank you. So it's in this building, room 8, so just down 1 floor. Group C and D will be starting with the NGSS activities in room 132, which is just on the other side of this wall, so you don't have to go very far. You can see the different options for the second workshop. Okay, so second workshops, there we've added citizen science, which would be in 152, which is just right across. You would go past the breakfast room and down the hallway, and you'll find it over there. Okay. Yes. Before that, we're going to take a 15-minute break. So feel free to get more coffee, get something to eat, and then try to get to your workshop room about 10:45. If you need our program, you can scan the QR code on your way out.
So can we if you want to go later? Or we have to stay?
No. You're here for the keynote. Yeah. So
Are we still gonna get the points?
send me an email that says that you were here, and you'll get the points.
Okay, I have to send you an email. Okay.
This is for Bio 105, right?
No, I'm in Bio 201.
Then you have to go talk to the instructor for 201.
The Dennehy.
He's here.
That was really very nice.
Oh yes. You might have to open Let's make sure it works. Perfect. Okay. So room 8, which is just down. It's not the usual lab that you go to. It's actually a little bit closer. Okay. So and okay. Oh, perfect. Oh, perfect.
Oh, that's okay. Okay.